Thank you, Hakan. Good afternoon. My proposal is titled Pharmacogenetic Analyses Across the International 100,000 Cohort Consortium by Means of Drug Response Assessments. As a theoretical and computational chemist, I'm fortunate to provide my knowledge and expertise in this subject. The definition of pharmacogenomics, as defined by the NIH National Library of Medicine, is to study how genes affect a person's response to drugs. It combines pharmacology and genomics to develop effective, safe medications and doses that will be tailored to a person's genetic makeup. Many drugs that are currently available are one size fits all, but they don't work the same way for everyone. It can be difficult to predict who will benefit from a medication, who will not respond at all, or will experience negative side effects, adverse drug reactions in this case, and are a significant cause of hospitalizations and deaths in the US. They occur in 10 to 20% of hospitals, and 10 to 20% of these reactions are severe. In this block of time, I'd like to go over with you the Center for Applied Genomics pharmacogenomic efforts, advanced or enhanced methods of pharmacogenomic data, the initial goal and objective, your approach to achieving them, cross-cohort and resource requirements. We made some impact in terms of pharmacogenomics and pharmacogenomic-related studies. Since making this talk, actually, we've done additional work on this subject. For example, morphine dose optimization for those with acute respiratory failure. What I'm showing you are a dozen or so studies that go from asthma, IBD, ADHD, and others. This search can actually be done through an academic search engine like Web of Science. From this body of work, we'd like to expand our approach that would address issues like the lack of specific and inconclusive data and prediction implementation issues. The first approach is deep learning and artificial intelligence. So deep learning is a variation of machine learning that uses multi-layer neural networks to automatically extract novel features from input data and capable of identifying highly complex patterns in large data sets. If you go to bioRxiv and search for deep learning, you'll see that there are more than 440 articles. Likewise, for PubMed, you'll see almost twice as much references. In that light, machine learning is an artificial intelligence technique that gives computers the ability to learn without being explicitly programmed to do so. And artificial intelligence refers to any technique which enables computers to mimic human behavior. The figure on the right there, and the video was kind enough to create a figure showing each other's linked to each other. You could also find out that Oracle and MATLAB created similar figures. Deep learning will be widely used to predict personalized drug response and optimize medication selection and dosing using knowledge extracted from large and complex molecular, epidemiological, demographic, and clinical data sets. So what I'm showing you here, deep learning consists of 3 layers: an input layer, a hidden layer, and an output layer, as well as their interactions to each other. The hidden layers define the deep learning concept. Now, if we assign variables to the input layer, defined here as x, and if we make the weight of the input node w, gamma here is defined as an activation function, which is normally nonlinear. In this case, we can make it Gaussian to transform this linear combination of an input signal from its respective node to an output value. This is representative of an artificial neural network and is done by an iterative modification of the weight in the network to optimize errors between predicted and true values, in this case of a backpropagation method. And there's the expression. We just have to remember that for a deep neural network, the hidden layers have to consist of more than 3 hidden layers. Right? From the information mentioned here, we're still able to, from the figure on the right, express a single layer network, finding a derivative, a partial derivative of the error, and calculating something called a loss function through the difference between the network and expected output. Now, of course, there are requirements of using deep learning for the sake of comparison to machine learning. One of the most significant differences is the time it takes to train and validate test data on a model. Here, I'd like to talk about renormalization and coarse graining as a second advanced enhanced method. Renormalization group and coarse graining is a fundamental concept in statistical physics. Renormalization is an iterative coarse grain procedure designed to tackle difficult problems in physics and now has been applied to data analysis, engineering science that involve multiple length scales. A central goal of renormalization is to extract relevant features of a physical system for describing phenomena at large length scales by integrating out short distances of freedom. Now, the figure I'm showing you represents a lipid molecule used to create phospholipid bilayers, representing a small piece of a cell membrane for the purposes of a deterministic molecular simulation. This figure also represents how renormalization can create various coarse-grained systems with respect to integration. So there are limits in computational power that are directly related to the size of the molecular system. The deterministic molecular simulation I was applying to is molecular dynamics here. It requires numerous calculations of energy of the system under study is at least of the order of this log function and can be prohibitive of large values of n. So there are two solutions to this. In the first one, we can modify the implementation of the software designed to perform these calculations, design a specific hardware tailored to the specific functions called by the software, and the design of new code that can harvest the power of this new hardware and newly available infrastructure. In this case, the graphical processing unit is a good example here and can increase the speed of a molecular simulation by several factors. The second solution is to modify the representation of the system and develop models with reduced number of particles to make the calculations more tractable. I was lucky enough to work on this problem during my my graduate tenure. So I'm highlighting this as a way to expanding this to large complex data sets. And the main difficulty in renormalization and coarse grading is to, in this case for molecular simulation purposes, design these potential energy functions and retain the physics of the explicit system in terms of structure, dynamics, and energy. As I mentioned before, application to large complex and heterogeneous data sets. And we have to keep in mind bias and information loss when we decide to renormalize and to create a coarse-grained system. This is a simple graphic. The top figure is a representation of data points in a grid, arguably a simple way to describe them. On the bottom is a renormalized and a coarse-grained system from using the top figure. So 0 prime is representative of the 4 boxes above. So far I talked about how to generate data using deep learning and how to analyze large amounts of data using renormalization. The final method I want to briefly talk about something called quantitative systems pharmacogenomics as an alternative and supplementary approach to the ones I mentioned here. So quantitative systems pharmacogenomics combines mathematical modeling that can consist of statistics, ordinary and partial differential equations to examine relationships between the drug, a biological system, and disease process. This integrates quantitative drug data with knowledge of its mechanism of action and predicts how the drug will modify cell signaling networks, how they impact and are impacted by human pathophysiology. The figure on the upper right-hand corner shows the impact of quantitative systems pharmacogenomics across therapeutic areas. We see metabolic diseases, autoimmune disorders, rare diseases, cardiovascular infectious diseases, cancer, and a large representation of neuroscience. Pharmacogenomics research has shown the diversity of patients' response to drugs. However, there are limitations of existing pharmacogenomic-based methods that used to predict a response to a particular drug and dosage combination. The approach will make medical treatment safer, more efficient, cost-effective by determining how an individual person responds to an individual drug by deriving and using sophisticated models, such as differential equations. We'll be able to simulate different variables a drug has in a patient, such as protein-protein or protein-DNA interactions. Specifically, the framework characterizes a drug's absorption, along with its distribution and elimination properties. This will yield information on pharmacological targets, physiological pathways, and disease systems in patients. In turn, this allows treatment's effectiveness to be predicted from these models. This will facilitate the quantitative prediction of individual subject responses, as well as the design of optimal drug treatments. From here, we can also determine what-if scenarios from these mathematical models. Now, the initial goal, objective, and approach proposal would be for 3 years. In terms of deep learning and renormalization, we will build and optimize, train, and test the data on the model. Once that is successfully done, a production run will be performed, and then following year 2 and 3, expanding to large, complex internal data sets and to cohort data. So the objective is to further elucidate and implement novel pharmacogenomics associations and trends from drug response assessments using these advanced and enhanced methods. Now, these dependencies out of our control can consist of compilation optimization errors, software hardware issues, local cloud issues, data analysis engineering science challenges, and they can consist of complex data distribution, missing data, measurement error, spurious correlation, and robust statistical method requirements. So, data and sample access of genomic data will be analyzed with an initial focus on genotype sequencing and targeted panels. The electronic health records derived data will also focus on the drug, dosing, allergy, and/or adverse drug responses. Now, sharing pharmacogenomic related data, especially generated from deep learning and/or renormalization, require a specific hub, a specific infrastructure. And for challenges, data requirements, overfitting, interpretability. The cross-cohort requirement, rationale, discovery, reproducibility, validation, and drawing activities which would bring accessibility, application, efficiency, and execution. The budget, the cohort is $1 billion for that time. Personnel would consist of an analyst, engineer, and/or scientist. The equipment can be a variety of CPUs, graphical processing units, tensor processing units, also high throughput performance and multi-threaded computing. Local and cloud clusters will also be implemented, as well as the option of using the extreme science and engineering discovery environment, XSEED, and the supercomputers that are part of that cohort, and relevant conferences every year for each analyst, engineer, or scientist. I'd like to thank those who made this possible: the International 100,000 Cohort Consortium, heads of international research organizations, Global Medicine, Genomic Medicine Collaborative, Global Alliance for Genomics and Health. I'd also like to thank John, Patrick, and Halkin, as well as everyone who made this trip to the summit. Thank you.
Thank you, Kenny. We have time for a question or two. Question, we gain 1 minute. Thank you, Kenny.